We will start the program from 2:30 p.m. onwards. So keep your patience.
হ্যালো, শোনা যাচ্ছে আমার কথা?
Yes, it's audible. Okay.
Okay.
Mhm. এই রে সুন্দরটা সত্যি হাসাহাসি করছে। Good afternoon, madam. ম্যাডাম, ডক্টর রে,
হ্যাঁ বলো। হ্যাঁ শুনতে পাচ্ছি।
ডক্টর একটু যদি আপনার স্লাইডটা একটু প্রেজেন্ট করেন তাহলে একটু
হ্যাঁ
টেকনিক্যাল
হ্যাঁ
ইস্যুটা
হ্যাঁ
চেক করে নেওয়া
হ্যাঁ
যেত।
আমি দেখে নিচ্ছি, এক সেকেন্ড দাঁড়াও, আমি একবার না লিভ করে জয়েন করছি। হ্যাঁ এক
ওকে
সেকেন্ড।
ওকে ওকে, কোনো অসুবিধা। ইয়েস ম্যাম।
হ্যাঁ আমি প্রেজেন্ট করছি, দাঁড়াও।
হ্যাঁ
শোনা যাচ্ছে কথা?
হ্যাঁ, ক্লিয়ারলি অডিবল। ওকে,
আমি প্রেজেন্ট করছি, দাঁড়াও। দেখা যাচ্ছে?
ইয়েস ম্যাম। এবার এটাকে একটুখানি মানে
স্লাইড
ফুল
ভিউ
স্ক্রিন,
করব?
হ্যাঁ ম্যাম একটু ফুল স্ক্রিন করতে হবে আপনাকে।
দেখা যাচ্ছে?
ইয়া ম্যাম।
ওকে, এভাবে রাখবো না আমি ছেড়ে দেবো?
আপনি রাখতে পারেন ম্যাম, অসুবিধা নেই। ওকে,
পরে করছি তাহলে, হ্যাঁ তোমাদের হয়তো ইন্টারাকশন থাকবে।
ওকে
আমি বন্ধ
ওকে
করি এখন।
হ্যাঁ হ্যাঁ ওকে ওকে, থ্যাংক ইউ।
Hello, Mr. Sarkar, am I audible?
Yes sir, you are audible.
Okay. I think some problem regarding my video is means my video is you can see.
No sir, video is not visible.
Okay, well, why I don't know, but it is coming when.
Kaushik sir, please unmute yourself.
But why my video
You are
is not
muted, sir.
coming? That I don't know.
I think sir, this is due to the technical issue, I think, a network issue, I think.
Hello.
Principal sir, you are audible.
No, my video is not coming. I don't know why. Hello.
This is network issue, sir.
I don't
Your
know.
audio is not clear as well.
Okay.
The network bandwidth is low, very low.
Okay. That may be. So I can
Yeah.
try. I try another one. Okay. So how many participants have joined till now?
It's 133, sir.
Okay, so it's a good number.
Sir, we should start the proceedings. It is
Just
2:30.
just
Okay.
1 or 2 minutes. Okay, so Mr. Sarkar, I think I'm audible and it is fine. I think all can hear me. Is it
Yes, sir. Yes, sir. Yes, sir.
fine?
You are audible.
Okay. No, so you told me that some voice is not coming properly. So that's why
No,
I
no, no. Now it is. Now it's
now it's
okay, sir.
fine. Now it's fine. Nah? So
Okay.
I think principal sir has also joined. So we can start our program. So I would like to. So Krishna ma'am also I think joined. Yes, Dr. Krishna Roy has also joined. So we can start our program. So before moving to the main session, so I need to clear one or two things from our technical team, that all the participants, their microphone, I think it is by default it would be muted, I think, and they cannot share their screen or these things. I think it is right, Mr. Musharkar?
Yes, sir. Yes, absolutely right.
Yes.
Okay.
So as we have given the time of 2:30, so we are going to start at I think now 2:32. So good afternoon to all dear participants, my colleagues, students, researcher, and obviously today's speaker, Dr. Krishna Ray from West Bengal State University. So I congratulate and I welcome all to this webinar and always being with us, because we have already organized 4 or 5 webinars from last 2020 to 2021, and all participants, they are really very spontaneously, they always supported us. So that's why I would like to congratulate and we are thankful to them. So today our theme is the magical microbes. So why we have chosen, being a pharmaceutical college, why we have chosen this topic, magical microbes? So it is quite, I think you can guess, all the audience can guess it, because after this 2020, that pandemic, the virus, the general people, even the science community, even the students, UG and PG level both students, they are only aware that virus can infect us, virus can cause the severe diseases, and they're actually harmful. But we are here to play a role. The being a teacher, being a scientist, we have to play the role. We have to transcend some messages to our society, particularly to the young generation, mainly focusing on the students, so that microbes always not harmful. Because very commonly we know that lactobacillus bacteria, they are very helpful for humans, and in treatment today's, in case of the clinical treatment, the lactobacillus strains are sold as a marketed drugs. So not only the good role of the microbes is not limited to this only, the microbes can play a major role in our today's society, as we are always focusing on the green society, green approach, sustainable approach. So here the role of microbes comes as a major player. So that's why we have chosen the theme magical microbes, to transform or disseminate the knowledge of microbial knowledges. How can we use that microbes in the different aspects, from the medical science to the agricultural science to the energy and other different sectors also. So with this, I think I can explain why we have chosen the magical microbes, this theme. So today with us, our first speaker, Dr. Krishna Ray. So before moving to the main part of lecture, I would like to invite our principal sir, Professor Dr. Amit Shankar Datto, to introduce our today's first speaker, Dr. Krishna Ray. Sir, please.
Hello, good afternoon and welcome, Dr. Krishna Roy, Assistant Professor in the Department of West Bengal State University. We have organized several seminars and webinars to enlighten our students, and so you are very experienced and work in different field as a research scientist, as a research associate also. So with your experience, our students and faculties both are enlightened, and I welcome you and ask you to start your speech. Okay.
Thank you. I first express my kind gratitude to the authority of Eminent College of Pharmaceutical Technology, and as they have given me this opportunity to present the magic what microbes can do, as already depicted by Kaushik. So I thank them from core of my heart, and I'm very much grateful to Kaushik, Dr. Kaushik Biswas, Associate Professor of this college, who actually contacted me, and he actually took the initiative to give me this opportunity. So thank you, Kaushik. And now I'm starting to share my screen. Just be patient. Can everyone see?
Yes, ma'am. It's visible. Okay.
Okay. So actually, I am not working on only this part. I'm working on various other parts where I can use the magic of microbes actually. But as Kaushik told me to present this part, so I'm presenting this part. So this is actually on biological phosphorus remediation from wastewater. I want to explain the scientific mechanism, how these microbes can be utilized to remediate phosphorus, basically from wastewater. We are dealing here only with wastewater. And is there any novelty in that? And how we can utilize this process for downstream useful uses. Okay, so I'm going to the next slide. So as you know, the phosphorus, it's the element which is very much essential for different life forms. You are very much aware that it is a component of DNA. It is a component of DNA, you know, and it's a component of cell membrane, of course. And the phosphorus, the ATP is actually the energy reservoir of our cells. So this is also basically made of phosphorus. But the source of phosphorus, it is a non-renewable one. You know, it's a finite source. From where we get it? We get it from the rocks, phosphorus rocks actually. But these rocks, these are finite sources. And the growing demand for this food, biofuels, all these things, the experts, they predict that due to this constant rise in use of phosphorus, this reserve may get exhausted by the next 40 to 400 years. Okay, so usually the soils, they have a very low concentration of phosphorus, but the problem is that this phosphorus is being utilized for several reasons. Basically, the highest use of phosphorus is in fertilizers. We know all around the world, fertilizers, the conventional synthetic fertilizers, they're being used in agriculture. And what happens, our farmers, they always use the fertilizers in excess of what they actually need, what actually the crops need. And what is the result of that? That these excess of phosphorus, these are all highly soluble. These fertilizers are very highly soluble. The phosphorus which is present in this phosphorus, it's very much highly soluble. And what happens? It comes as an agricultural runoff in the surface water, in the ponds, in the lakes, in the rivers, wherever it is. And then that causes the major problem. And we can see that there is a constant loss of phosphorus. And also this finite resource, it is very evenly dispensed around the world. Only Morocco has 85% of the global share, followed by China with 6%, and US also, it has around 3% something. Next slide. Okay, so this is as a whole, we can see this is the actually the scenario of global phosphorus use. If we start from the rock phosphate, collection of rock phosphate, then we make it available for human uses. It is mainly used for agricultural uses, and then it is used for food supply and for several other uses. But at the same time, there is also losses. There is also losses from this use, and very small amount is actually recycled. So this waste treatment, and by this wastewater, if it is treated and if this phosphorus can be recycled, then we can make this finite resource, which is very non-renewable resource, we can make it reusable, recyclable, and renewable. Okay, and you can see this loss can cause a lot of problem in the aquatic life forms. Okay, it can cause several other problems also. I'm citing this. Okay, then according to whether is this phosphorus, is it toxic, or why we are so concerned that this phosphorus, one reason is that it's finite resources. Okay, but if it gets disseminated in the environment, does it cause any problem? Why we are so concerned? Actually, in our country, there is very less concern about the phosphorus toxicity, whether in soil or whether in water, it's very less. But if you see the international journals, I'm citing one journal particularly. This is environmental, EST, this Environmental Science and Technology. This is published by American Chemical Society. It's a very high impact factor journal. You can see the publications there. It is actually, the researchers are expressing quite concern about the high level of phosphorus coming into the environment as a pollutant, and how this phosphorus can be recycled. They're really worried about how it can be recycled, how this toxicity, this pollution can be remediated, all these things. So it's really a major concern. And is it toxic? So according to US Environmental Protection Agency, EPA has classified phosphorus as a group D element, which is not causing any carcinogenicity. Okay, and white phosphorus is actually, it causes problems. It is actually causing problem, the white phosphorus. It is extremely toxic, but other forms of phosphorus, they are much less toxic. And we all know that phosphorus is applied as fertilizer for crops, but for crops also this phosphorus can be toxic. If it crosses the critical value, each and every crop, it requires a certain amount of phosphorus, beyond which, if it is applied in soil, instead of causing better growth, better yield of the crops, it might lead to crop failure. When I started searching for, is there any report of phosphorus toxicity in our country, I found a report that by Indian Institute of Spice Research in Kerala. They said that there were high level of phosphorus. It's far greater than 100 kg per hectare, which is the standard advised phosphorus for a particular crop. They are supposed to describe these things for spice research, so it might be the spice research actually. So it was very high beyond the critical value, and it led to crop failure. And subsequently, our studies also, we made a small study in our laboratory environment, and that also led to the conclusion that if we use phosphorus higher than the critical value of the crop, that is actually causing problems. That is actually inhibiting the growth of the crop rather than accelerating its growth. So really, higher level of phosphorus is really toxic for plants. And also we know that if excess phosphorus accumulates in water bodies, it causes algal bloom. It is very much, we know that it's a very common term, and it endangers the aquatic lives. And for human beings also, I have already told you that white phosphorus is particularly toxic. And also, if it is used in, if it is human being gets exposure to higher than a certain level, in low level, it's not a toxic element. Okay, so there is much concern that if this element it gets unnecessarily disseminated in the environment, it might cause problems for the life forms. Okay, and for wastewater, as we are concerned here with wastewater, so municipal wastewaters, they do not have very much amount of phosphorus. It's generally 5 to 20 milligram in total. It may be out of that, certain may be organic phosphorus, certain amount may be inorganic phosphorus. And detergents, the detergents we use for cleaning our clothes in washing machines, these are the major source of phosphates in the environment in the form of domestic sewage water. But there are some industrial effluents which are actually very high in phosphorus level. And there comes the problem, whether those effluents, they can get rid of the phosphorus before they are discharged into the environment. So what are those effluents? They are fertilizer industry effluent. It has very high, it's reported to be very high phosphorus containing effluent. It's 25 to 308 milligram per liter. Then in our research and several other research across the world, this parboiled rice mill industry, the parboiled rice mill effluent, it is also very much rich in phosphorus. 34 to 143 milligram reported from Brazil, from US 98 milligram per liter, and in our publication, we have cited it within 4 to 72 milligram per liter in West Bengal. But there are several other reports across India, so many reports, and they also, sometimes they are greater than 100 milligram per liter. Okay, and also the rice-based distilleries, these are also very much rich in phosphorus. And what is the permissible level of phosphorus discharge to the environment? WHO, they have fixed the level of phosphorus to be 0.1 milligram per liter in drinking water. And in US, the effluent limitation is phosphorus effluent limitation is equal to 1 milligram per liter for total phosphorus. And what about our country? In our country, the pollutants amount, it is allowable pollutant amounts, it is actually decided by the Central Pollution Control Board, CPCB. And I had to really search for the CPCB documents for finding this permissible limit. So I found that for dissolved phosphates, soluble phosphates, in the effluents which will be discharged into the inland surface, there is a limit that is 5 milligram per liter. Okay, so this is the actual scenario for the permissible limits of discharge of phosphorus into the environment from the wastewater effluents. Okay. Now, what are the major processes by which this wastewater can be made free of phosphorus? Is it possible to make it free of phosphorus? So there are several processes. What are these processes? There are several processes. So this is in a nutshell. You can see primarily it is always done in a settling tank. It is allowed to, the wastewater when it comes, it is allowed to settle. Okay. Then from that tank, it is after settling this way to wastewater, it is passed through secondary treatment, tertiary treatment, all these things. And the major part of phosphorus which is removed, this is in this tertiary, secondary treatment only. This is major part of the phosphorus. It is removed in the secondary treatment, and then it is followed by tertiary treatment. So tertiary treatment, it may be, basically it may be physical treatment. It can be chemical treatment. Okay, but secondary treatment is the major treatment through which the phosphorus is actually removed from wastewater. And out of this, the red colored treatments, you can find this constructed with plants, biological filter, conventional activated sludge. These are the treatments which are followed in our country also as well. And these treatments can be performed in decentralized systems also. Decentralized system is in small scale. You can do it in small scale systems also. It's equally applicable. But this enhanced biological P removal, this is EBPR, this is it is reported to be the most cost effective system. It has to be performed in a centralized system. And the effluent which is passed through this system, after that it can have tertiary treatment also with the chemical precipitation, all these things. And algal system, algal bacterial hybrid or sole algal system, it has become very popular for removal of phosphorus from wastewater. Nowadays, if you find out this related journals, you can see there are several publications, good research publications, where these algae, the potential of this algae for removing phosphorus from the wastewater, it is highly exploited. It's exploited in a very, very large scale. So algae is a good, it's prospective good biological organism. Next to bacteria, which is used in enhanced EBPR, this EBPR system, next to that, algae is another preferred biological organism which is chosen for this phosphorus removal from wastewater. Otherwise, chemical treatment of phosphorus is very well known. You just have to add calcium carbonate. You can just add, and that will give massive precipitation of phosphorus. But the problem is, after you get the precipitation, your wastewater will be free. Of course, the wastewater will be free from phosphorus, but the precipitate, the sludge which you will be developing, that sludge, how will you dispose of this sludge? This sludge will again, if you dispose it in the environment, that will again create in the creation of phosphorus pollutant. So that's the problem. So it's better if we can recycle it through the biological organism. That would be much better option. So this EBPR, so what our center of study, it will be EBPR. What will be this EBPR has a potential to remove phosphate by 80 to 90%, and if treated effluent, which will come out from the EBPR process, it can be even as low as less than 1 milligram per liter. It's usually, it is like this. But we have to remember that when the maximum removal efficiency of EBPR is observed when the influent phosphorus level is very low. Influent phosphorus level, it has to be very low. Then only you will be able to get the maximum efficiency of the EBPR. For example, 93 to 95% of phosphorus removal is reported from EBPR in municipal wastewater. This is a report from China, two reports in 2001, 2011 and 2017. What they say that influent phosphorus level, it's around 3 or 8, something 3 to 8, something like that. It's very low. And then only you can get the effluent level even below 1 milligram. It's 0.25 milligram. You can get the effluent level. But we have to remember that the influent level of phosphorus, if it is low, just like municipal wastewater, usually municipal wastewater, it has very low phosphorus level. So for this EBPR system is quite perfect. But the problem lies when we want to purify this high phosphate rich wastewater with the help of EBPR. So is the EBPR that much effective in removing phosphorus from phosphate rich wastewater? Let us see. So what is the principle? What are the principles behind this enhanced biological phosphorus system? This is phosphorus removal system, EBPR. Actually, in EBPR, the magic, what Kaushik said, that magic of the microbe is actually utilized. How? It is an activated sludge process where this polyphosphate accumulating organism, this PAOs, the efficiency of the PAOs is being exploited. What do these PAOs do? They take up the soluble phosphorus from wastewater and they accumulate it in the form of polyphosphate. This is insoluble phosphate, polyphosphate granules. And this phosphorus uptake it is enhanced in EBPR system when these organisms, they're alternated between an carbon-rich anaerobic environment and carbon-rich anaerobic phase, and another is carbon-poor aerobic or anoxic phase. In aerobic phase, oxygen is the electron acceptor, so we call it aerobic phase. And in anoxic phase, nitrate or any other gas, that can be the electron acceptor. So if it is alternated, these PAOs are alternated between an carbon-rich anaerobic phase and a carbon-poor aerobic or anoxic phase, the maximum efficiency of their P uptake ability that can be exploited. And what is the property of POs is exploited here? That is the luxury P uptake. What is this luxury P uptake? These POs, they can utilize they can uptake this phosphorus. It is in excess of their required amount. They do not need so much of phosphorus for their to perform their metabolic activities, but still they have a tendency to uptake this phosphorus and they accumulate it inside the cell and they preserve it in the form of what? In the form of polyphosphate granules, poly-P. I'll tell you in detail about this, okay, later. Okay, so current understanding of this biochemistry and microbiology of this EBPR is still incomplete. Sometimes it is quite enigmatic for downscaling and upscaling treatment approach, but people are trying. It's an ongoing research. So what I told you, it's in a detailed manner I'm showing you. So you see what happens in anaerobic condition. What happens? One carbon source has to be used in the wastewater. We add this carbon source for EBPR system. So this is VFA. VFA is the volatile fatty acids. It has been used as a carbon source here in EBPR process, and this VFA, well, the organism, they take VFA, they convert it to their through TCA cycle, they ultimately they convert it to PHA. PHA is polyhydroxy alkyonate. This PHA granules, this polyhydroxy alkyonate, these are also another compounds which gives energy to their hydrolysis also supplies energy to the bacteria. So in the anaerobic phase, they actually utilize this VFA, and from this VFA, they form this PHA. And for formation of the utilization of or carrying out of this whole process, they utilize what? They utilize some of the poly-P granules already stored in their cells, inside the cell. The polyphosphate granules, which are already stored inside their cell, these polyphosphate granules, they are n numbers of phosphates which are being bound to each other with a phosphoanhydride bond. And this phosphoanhydride bond is very much rich. It's very energy rich, these phosphoanhydride bonds. And so upon hydrolysis of these phosphoanhydride bonds, a large amount of energy is actually released. So what did they do in the initial anaerobic phase? This VFA by the organisms is utilized to form this PHA with the help of hydrolysis of poly-P granules which are already stored inside them. And hydrolysis of poly-P leads to formation of free P, which is actually diffused out of the cell in the wastewater. So instead of taking phosphate from the wastewater, in the first phase, the organisms, the PAOs, the polyphosphate accumulating organisms, they actually discharge phosphates in the wastewater. So this is the first phase. And the next phase, they are alternated. It is followed by the next phase where no carbon source is used. Here, no carbon source is added in the wastewater. Instead, they are supplied either oxygen or they are treated with nitrate or nitrogen gas, whatever. And then in that case, what happens? This PHA is being utilized by the organisms for their increasing biomass and their performing their metabolic activities. This PHA is used, and at the same time, what do? They take up a large amount of phosphates from the external environment, that is the wastewater. So at this phase, no carbon source is added, and instead a large amount of this PHA, which is which was synthesized in the earlier phase, that is utilized, and a large amount of polyphosphate is synthesized here by uptake of phosphates from the wastewater. So in one phase, phosphate is being released in the wastewater, and the next phase, the phosphate is being absorbed by the, not absorbed, we can say accumulated, it's taken up by the microbial cells. Okay, but what happens, the net uptake is always higher than the net release. Always, it has been found that the net uptake is always higher than the net release. So ultimately what happens? Ultimately, it results in the biological wastewater phosphate removal. So ultimately, this biological wastewater phosphate removal happens with its product as polyphosphates. So keep this in mind. I'll go to it in the next phases. So this is the EBPR process. This influent is anaerobically done, then aerobically done, or anoxically done, whatever they prefer. And here the activated sludge is actually used. No additional microbes are being added. The microbes which are present in that wastewater itself, that is being utilized. The polyphosphate accumulating organisms which are present in that sludge, that is actually being used, and ultimately it is clarified and used as an effluent. Okay. So what are this in this sludge, these EBPR sludges? What are the organisms present which are actually responsible for such high amount of polyphosphate accumulation or such high amount of phosphorus uptake? So far, it has been identified that Accumulibacter phosphates, this is the most dominant POs in the EBPR system. Earlier, Acinetobacter was supposed to be dominant EBPR, but later this it was disproved that they are obviously Acinetobacter species are polyphosphate accumulating organisms, PO, but they are not major component in the sludge of wastewaters. So major component is Accumulibacter phosphates. In addition to that, tetrasphaera related actinobacteria, they were also found to be quite prevalent components of. They are also present in quite high abundance in these EBPR sludges. And in addition to that, Pseudomonas, Paracoccus, Enterobacteria, these were also reported to be the prevalent PAOs found in the EBPR system. But but what happens? In addition to this, there are certain POs which can perform both the phosphorus accumulation as well as they can do denitrification also. Okay, they can they do not need the aerobic phase, rather they prefer the anoxic phase where nitrates can be used. So they can at the same time they can do both the denitrification and phosphorus removal. Both can be done, and this group of organisms, they were termed as denitrifying POs or DPAOs. Okay, they use nitrate as their terminal electron acceptor for the oxidation of PHA under anoxic condition. So it has been suggested by several researchers that if DPAOs can be utilized in wastewater treatment plants, then we can save a lot of energy, because it will give the two benefits. It can denitrify the water. If wastewater is equally rich in nitrate and phosphates, it can be purified, it can be bioremediated at the same time by these DPAOs, because the nitrates will be denitrified by these DPAOs. At the same time, phosphorus will be uptaken by them. So these ideas, they are also being exploited. But do you think that however rich, higher amount of phosphorus will be present in the wastewater, the POs will be able to take up, they will be able to accumulate phosphorus from that wastewater? Actually, the answer is no. there is a limit of these PAOs. Usually it is observed that at lower external concentration of phosphates, these PAOs, they remove phosphorus with very high efficiency. But as you go on increasing the phosphorus level in the wastewater, at one point of time they get saturated and they cannot take phosphorus from the wastewater beyond a certain level. And this certain level, if you ask me what can be the certain level, is it common for all the POs? Actually, the answer is no. It differs from PO to PO. So, and it depends on the condition also under which it is absorbing phosphorus, it's taking phosphorus. So phosphorus uptake is basically, it is done by the PST. These are the phosphorus specific transporters. These are very high affinity transporters which are present in the cell membrane. So these cell membranes, they actually uptake phosphorus from the outside environment and they send it to the cell. So this PST, expression of these PST genes, they are actually responsible for higher uptake of phosphorus. And it has been found, it has been already, the idea is there in from E. coli, extensive studies in E. coli, that there is a regulatory protein, its name is 4U. This acts as a negative regulator of this phosphorus uptake. They actually, this 4U is known to repress the expression of these phosphorus transporter genes, it's PST genes actually. So this 4U is known to be a negative regulator of phosphorus uptake. So we cannot expect that if we increase the amount of wastewater, phosphorus in the wastewater, the EBP in the POs will go on accumulating more and more and more phosphorus. Actually, it doesn't take place. At that time, the 4U comes in the way and it acts as a negative regulator of phosphorus uptake. So that's the problem. And the phosphorus uptake, total phosphorus uptake system, it is actually, it is controlled by the 4-regulon, and it is extensively studied in E. coli. And this 4-regulon consists of two component system genes, this two component system proteins, these are 4R, 4B, along with 4 PST transporters, PST-S, PST-C, PST-A, PST-B, along with the 4U, which is known to be a metal binding protein. And in E. coli, it has been found that when there is very low concentration of phosphorus, the 4B is activated by 4R as a kinase, to act as a kinase, okay? But under phosphorus rich condition, this 4B is interrupted by 4R as a phosphatase. And what is the role of 4U in this? 4U actually, it helps the 4B in this dephosphorylation to act as a phosphatase. The 4U is intimately involved in dephosphorylation activity of 4B, and therefore what happens, this uptake of phosphorus is actually interrupted. Uptake of phosphorus will be interrupted when these PST genes, the phosphorus transporter genes, they will stop acting. When they will stop acting, when this 4B will act as a dephosphorylating agent. So therefore, this 4U, as I earlier said, that it is known as a negative regulator of phosphorus uptake. So people started to use the mutated form of 4U to get whether this 4B can be a constitutive kinase. They can be utilized as a constitutive kinase, whether is it possible or not. These ideas people have started to use long back. But what happened, the mutants, 4U mutants, they initially they showed that they can take up high amount of phosphorus, but easily they are outgrown by the revertants. Revertants means in which the mutation is again rescued. Okay, so gradually it was found that they get outgrown by the revertants, and the mutation actually it becomes ineffective. So is there any way so that we can overcome the regulation of 4U, negative regulation of 4U, and make the cells like that so that it can take on more and more phosphorus? Is it possible? So that was the first thing which we were thinking when we started our work. And you can be having one question, that why we were interested in biological phosphorus uptake, because we found that the phosphorus ultimately it is taken inside the cell as a polyphosphate granules, and we wanted to use these polyphosphate granules as biofertilizer. And one such fertilizer which is formed as from this wastewater, this is called struvite, but this is not biological precipitation, it's a chemical precipitation process where in this struvite there is also some amount of nitrogen and magnesium is also there, but this struvite is recovered from wastewater, and it is now known as a good alternative to the phosphate fertilizer, and it's known to be a very slow release phosphorus fertilizer, and it is achieved from wastewater treatment only. So we were interested whether we can make something like that, recycling through the biological recycling of phosphorus. Can we use these polyphosphate granules as biofertilizer for crop plants? So we expressed first this thought in this environment science technology. It was long back in 2013. Then that time we just started our work, and we wanted to go in this process. And that time we started working with the cyanobacteria and microalgae. And this is the structure of polyphosphates. You can see this is the structure of polyphosphate. You can see the energy rich phosphoanhydride bond. It actually makes this polyphosphate granules. It's n number of phosphates, they are attached with this phosphoanhydride bonds. This is very energy rich bond. And so this bacteria, cyanobacteria, as I have told you, that it actually assimilates phosphorus in the form of poly P. And what is the significance of this poly P? That it can act as ATP. We know the ATP by its hydrolysis, its energy-rich phosphate bonds, it supplies energy to several metabolic activities of our life, of our body. And like that, in bacteria also, this poly P is a major source of energy, and they also help the organisms to adapt to extremes of salinity, osmolality, desiccation, UV radiation, pH, temperature, whatever. There are several abiotic stresses which can be overcome through the help of this polyphosphate. So they are important component of microbe system, bacteria, microalgae, or cyanobacteria, all of this. So when we plan to utilize this polyphosphate from wastewater, which will be rescued from wastewater, we first had to design how we can extract this polyphosphate and can we quantify this polyphosphate. So we have been successful in this process. So ultimately this we published this method article. It is very much highly cited article. And then whether it can be observed, we can visualize. So this was also done by Chandan. So this by the use of DAPI, people used to do it earlier also. It's an improved method. We made it highly reproducible. We made the method highly reproducible for cyanobacteria and microalgae. So these two methods we published in Protocol Exchange. And how does it look? How does the polyphosphate look? You can see this is the very basic staining which we used to do. That was Albert staining. Just we used to use toluidine blue, and here the darker colored cells, these are the polyphosphate accumulating cells, and these are all microalgae. These are all microalgal cells. Then we did the TEM, transmission electron microscopy, made the sections, and here you can see these are the black these granules. These are actually the polyphosphate accumulating. These are the polyphosphates. It actually under scanning electron microscope, transmission, obviously transmission scanning electron microscope, it looks like this. And after DAPI staining, how does it look? These two panels are from the light microscopes, and these two panels are from confocal microscope by the use of DAPI stains. You see the fluorescent, this green color, these actually shows the presence of polyphosphate granules, whereas the green, the blue colors or the light green colors, they actually refer to the non-polyphosphate accumulating cells. So this much we could do. We couldn't do it for bacteria because bacteria is much smaller. These are all for algal cells, but for bacteria, they're very much smaller. And in confocal, the whole of the bacteria used to get stained. So we could not differentiate between non-polyphosphate accumulating bacterial cells and polyphosphate accumulating bacterial cells. That we could not do at this moment. We are not able to do that. Then we identified, as we have already told you, that there are several phosphorus rich wastewater effluent. One of them is parboiled rice meal effluent. So we started with that parboiled rice effluent, and these are the setups. These are very indigenous setups. These are setups made in our greenhouse with just aquarium system and with light hovering over this. We started purifying the phosphorus, taking bioremediating phosphorus with several microalgae. This is the system. It was all set up by Chandan, my PhD student. These are all his ideas. He started working like this. This is a very indigenous system from where we started to see whether it is possible to harvest in a laboratory scale, whether it is possible to harvest the phosphorus itself as polyphosphate granules inside the cells, be it bacteria or be it microalgae. We tried both the systems, and all these microalgae and bacteria, they were native to this parboiled rice mill wastewater. We isolated all these organisms from the wastewater itself, because we do not want it to use any exogenous sources, biological organisms, because it will just replace the activated sludge actually. So we wanted to use the native organisms. So this is the very indigenous system which we set up. This is, you can see this parboiled rice mill also, its effluent is also very highly colored. You can see it's almost brown. Okay. And what happens? This is the observation, up to 20 days observation when we are growing the algae in this parboiled rice mill effluent. Gradually the color also gets diffused. There are several other parameters. Actually, this parboiled rice mill is very highly polluted. It has very high BOD, COD, high color. There are very less DO amount. DO is almost near to 0. Okay. So all these things get improved when we treat them with these organisms, these microalgae and this native bacteria, which we have isolated from there. But I'm not going to the details of this. I'm just concentrating on the phosphorus. So we found that they are growing very well, and ultimately they get aggregated when the microalgal cells and the filamentous algal cells, they are cultured together in this wastewater. They form aggregations, and it becomes very easier for us to separate, because when you are using microalgal cells, only microalgal cells, then it becomes the coagulation of them, removal of them from the wastewater, it becomes another problem, because I have to harvest. My purpose is to harvest the organism which I have been growing in the wastewater. So how can I harvest them? If it's not possible, if they are very small, how can I harvest them? So it has to be harvested by using natural coagulants, or it itself, it should get aggregated. So for microalgae and filamental algae, when we cultured together, we found that there is no problem in their aggregation, and we can easily remove them from the wastewater as a harvest. For bacteria, obviously we use several natural coagulants. Later, I'm not going to detail into that, but we use natural coagulants to separate the bacterial mass after culturing them in this wastewater. So, and after harvesting the mass, we used to dry them, we used to weigh them, we used to measure the polyphosphate content in them, and then we used to apply them for this rice seedlings. This much we could do in our greenhouse, and we tested the soil for release of the phosphorus from this harvested biomass. When we applied it in the soil, we tested regularly, on a regular basis, we tested the release of phosphorus from in this soil. And we also optimized the equivalent biomass of the organism, which is equivalent to a certain phosphorus level of the conventional phosphorus, conventional fertilizers, this NPK and superphosphate. Both of these we used, and which amount of superphosphate or what amount of NPK is equivalent to the biomass of the organisms which we will be using, that also we have optimized, because we have tested there, we extracted polyphosphates from there, and that polyphosphate is equivalent to how much phosphates present in the conventional fertilizer. That also we optimized, and depending on that calculation, we added this into the soil. We set up several experiments, and the interesting finding is this. You can see that this NPK is this purple line, and up to 115 days we have tested, and we found it is almost equivalent, this release of phosphorus from this NPK and release of phosphorus from these biological harvests. These are almost similar. This was our 2015 paper where we have shown this equivalence. We have established this equivalence later, and we found that the thing is that this NPK, it releases phosphorus, and all of them, they have a peak at 45 days. Upon that, after that, the NPK, the conventional fertilizer, it started to its availability is becoming lesser and lesser in the soil, whereas the biological organisms, they still maintain a good level of phosphorus in the soil. So when this is going down, this conventional fertilizer is going down, then biological fertilizer, it can make the steady release of phosphorus. So if we combine these two, it will be helpful. Then we can use a less amount of phosphorus also, the conventional fertilizer also, if we use both of them in combination. So these are just the beginning, just a hunch and just a beginning, which we started to get. And then we did it in a larger manner for algal strains. And here we used an algal consortium. We used both sterile soil and non-sterile soil. In non-sterile soil, we found that there is no release. In sterile soil, we found no release of phosphorus. That means in non-sterile soil only the release is taking place, and it must be assisted by the phosphorus solubilizing organism. The PSBs, PSOs, phosphorus solubilizing organism, they must be actively taking part in release of this phosphorus. Without their help, it's not possible. So that also we established that in our paper, because in sterile soil, you can see there is no release of phosphorus. Okay, but in whereas the conventional fertilizer is well released, this superphosphate and NPK, they are being released. But whereas our algal consortium, it failed to release any phosphorus in the sterile soil. But when we use non-sterile soil, we can see very interesting phenomena, that the release of phosphorus from the soil is from the biological organism is slow at initial phase, but gradually it increases when the conventional release of phosphorus from the conventional fertilizer is already reached a plateau. But here it is gradually increasing. So it's a slow, steady release, which is actually eco-friendly. Excessive release and rapid release is never desired actually for its becoming discharged in the environment. So it's never expected. And what about the growth parameters? We couldn't do much with the growth parameters, whether the addition of this algal consortium or bacterial consortium, whether it is better as a fertilizer than the conventional one. We cannot say that too much of data has been taken. It's only in greenhouse experiment and only what we could do with shoot height and leaf width. And there we show that there is a significant equivalence between that, the compatible, both are compatible, the algal biomass or bacterial biomass and these superphosphates and NPK. The levels are, their effect on the shoot height and leaf width, it is compatible. They are giving more or less same Then we started working on the next phase. Next phase means whether it is possible to overcome the negative regulation of FOU, whether it is possible. So this is the paper, last published paper by us from this stream in 2019. This is, we published this when after Chandan left because it had too many data and it took a long time to be organized. So what we did here, we isolated 13 bacterial isolates from this purple rice meal effluent, and we found that all these bacteria, when we sequenced their 16S RNA gene, we found that they all belong to 3 genera: Bacillus, E. coli, basically Escherichia coli, not any other Escherichia, and Staphylococcus. They only belong to these 3 genera. And one important biochemical character which is common to all these organisms, that they were all positive for nitrate reduction, and at the same time they were also having the ability to accumulate phosphorus. So we can call them as DPAO, denitrifying phosphorus accumulating organisms. We can call them because they can all denitrify at the same time they can accumulate phosphorus. So whether these DPOs have any role in overcoming the FOU negative regulation, that was the thing which we wanted to see. And for this study, we used effective control, which at the control, this control was on non-PAO bacteria, which this is the standard E. coli K12 strain, 2 strains of K12 strain we used, and E. coli DH5 alpha strain we used. These are known to be established non-PAO bacteria, but they were also denitrified. So these are also denitrifying, non-PO, denitrifying but non-PO. They are known to be denitrifier strains but non-PO. Okay, so these were used as a control, and I'm showing you what results we got. So initially, you just see that I have already told you that if you increase the level of phosphorus in the outside environment, what will happen? Gradually, it will stop absorbing, uptaking phosphorus, and it will gradually go down. It will gradually go down. So, okay, Kaushik is giving me, asking me to finish it quickly. So I'm finishing it quickly. So you see that in these PAOs, as we are increasing the phosphorus level, it is already, you see the phosphorus removal level efficiency is going down. It's applicable for both non-PO and non-PO, and non-PO and PO. All of them, you see, it's 100% at low level of phosphorus, 10 ppm when we are using, but when we are using 1000 ppm of phosphorus, it's going down. But we found that when we are using nitrates in the external medium, suddenly it went up. At 50 ppm of nitrate, it's not going up. But at 2000 ppm of nitrate also, it's not going up. But within 100 to 1000 ppm of nitrate addition, it's going up, and it is enhanced too much from, it's very much enhanced, you can see. And all the POs we isolated, they were all showing almost 80% of phosphorus removal, even irrespective of the concentration of phosphorus. We found it till 1000 ppm, it is taking phosphorus with 80% efficiency. Okay, whereas with nitrate we found at the same time it is removing a nitrate at 100%, both non-POs and POs, because both of them are denitrifier and there is no effect of external phosphorus on them, and it was removing nitrate at 100% efficiency. And at the same time we found that corresponding increasing level of polyphosphate accumulation. Okay, and this is the last one. here we studied the corresponding gene expressions. What is how this with nitrate, why this phosphorus accumulating efficiency it's going up? Why after not why, we can see what is actually the expression level of the corresponding genes. What we found that, you see, this is the expression level of these PST transporters and this is the expression level of 4U. So when at 0 phosphate, when there is no phosphate, you see this PST gene expression is highest, whereas 4U expression is lowest. But as we increase the phosphorus level, we have used here two phosphorus levels, 50, 100 and 1000, 500, 50, 100 and 500. We see that at 50, just after adding 50 ppm phosphorus, the PST expression level is going down, whereas suddenly the PST 4U expression level, the negative regulator level, is going up. Whereas when we added along with phosphorus is added nitrates, when we added nitrate, we found that gradually at initial level the 4U expression is high, but gradually this 4U expression is going low. It's going low, whereas the PST expression level is going high. So somehow then we observed that within 100 to 1000 ppm of nitrate addition, this enhancement occurs, but at 2000 ppm of nitrate addition, it doesn't occur. Okay, so the negative regulation of 4U can be withdrawn with the application of 100 nitrate addition. Okay, so but we could not at this level we cannot speculate, we cannot say what exactly is happening. So we speculate there must be some crosstalk between this 4-regulator and the nitrate signaling system. Nitrate signaling is bacteria also taking place with another 2-component system. So might be there any cross connection between this 4-regulon gene expression and this nitrate signaling. So, but it is still at speculation stage. I do not know actually what is happening. So this study is open, anybody can work on it. And initial this acknowledgement, this initial part was financed by one ICR project, and it's initial part, but the rest of the part we did our with their own funding. And these are the people who are the key persons behind this work. Obviously Chandan is it was his PhD thesis, so he was actively involved. These were all his ideas, he planned his experiments, I just helped him, but otherwise it was totally his ideas, his innovations, everything was his. And Rajajit was a key follower of Chandan. It was not his PhD problem, but he was intimately associated with Chandan to make it a success. And there is, it will be very much ungrateful for me if I do not mention the name of Mumtaj. Actually, when our last paper, Frontiers, it came back with reviewers' comment that we have to redo some RT-PCR expression of these bacteria. So that time Chandan has already left, and Rajajit didn't have much experience in this RT-PCR work, but Mumtaj has done it for plant samples, and Mumtaj has come forward and he has taken up, with bravery, he actually did all the experiments, redid the experiments with Rajajit, and then subsequently this paper got accepted. So we are very much grateful to Mumtaj, because it's not neither part of his thesis, her thesis or whatever, she didn't have any experience, but she pitched in with a lot of courage. So thank you. Sorry, Kaushik, I took much of your time. Okay.
No, ma'am, it's fine. So I'm really thankful to Dr. Ray for this beautiful presentation, very descriptive presentation, and which is very applicable, and I think it is completely matched with our theme, the magical microbes. So I think through this presentation, ma'am already depicted the theme, the magical microbes. I think our participant enjoyed. So there is one question from Coel Paul: are polyphosphates harmful?
So far as I know, it's not harmful. So far as I know, it's not harmful, but as already reported by medical clinical people, that the white phosphorus is harmful, but for polyphosphates as such, I do not know anything about being harmful.
Okay, ma'am. So there is no more question, but really we enjoyed the session, And I'm really thankful to ma'am with when I approached to madam, she come up with very readily and spontaneously. So I'm really thankful, and I hope that we will organize more webinar where madam will talk about the applications of this bioremediation of phosphorus or any more. So with this, I would like to thank madam from our college, Eminent College of Pharmaceutical Technology. So we will move towards the second session. I think Dr.
Thank you so
Kosturi
much,
Datta from NIT Rourkela is already joined. So I will hand over the session to our assistant professor Shinjini Das Chakraborty to conduct the second session. So Shinjini, can you hear me? Shinjini, can you hear me? Hello.
Hello.
Yeah. So I would like to hand over the program to Shinjini.
Okay, a very good afternoon, one and all. Not to waste much of the time, I would directly come to the second part of our session. So I would like to invite our beloved principal sir, Dr. Amit Shankar Datta, to come and give a small introduction about our second speaker for today, who is Dr. Kasturi Datta. So sir, Amit sir. Dr. Amit Shankar Datta, is he there? Sir, am I audible, sir? Hello? Am I audible?
Yes, you are audible. Yeah. Shinjini, you are audible. Principal sir, please.
Principal sir, can you please give us a brief introduction about our second speaker, Dr. Kosturi Datta?
I think, sir, there must be some internet issue over there. So Shinjini can continue it.
Okay, so I would like to invite our second speaker, who is Dr. Kosturi Datta, Assistant Professor, Department of Biotechnology and Medical Engineering, NIT Rourkela. She'll be here today to give us a small talk about the topic and overview of bioenergy as an alternative source of energy. So that sounds a very interesting topic. Ma'am, are you there?
Yes, hello.
Okay, I'll hand over the forum to you, ma'am, now. Please continue.
Yes, very good afternoon to all of you. And first of all, am I audible clearly?
Yes, ma'am.
Yes, ma'am, you are audible.
Okay, thank you. So first of all, I want to thank Dr. Kaushik Bishwas for inviting me for this webinar. And today, just start. Yeah. So I'm going to start my presentation with the topic that bioenergy and sustainable and renewable source of energy. So coming to the interaction about the bioenergy, so more or less all of us is aware of that fact that our recent energy resources, which is mostly the fossil fuel, is a perishable one, and for a lot of loading is there. Okay, large pressure is there on that stored fossil fuel, and if you don't want or don't find any another option of that, then in the near future it will find a severe lack or the energy crisis. So in the introduction, I want to introduce this energy sector as the most elementary requirement for the development of the whole world, okay, of the our nation and the world. And we generally use the traditional energy resources, that is the fossil fuel, and it has several issues recently which is of our concern, that is non-renewable source, and may cause the energy crisis and also cause the climate change due to longer use of this fossil fuel energy. It will release a lot many greenhouse gases emission in the environment, and that will affect our environment adversely, and it cause the climate change. Also, the release of gases like nitrogen oxides and sulfur oxides while burning this fossil fuel, this affect the environment as well as the human health and the whole ecology. So it's already known that right now, whatever the demand we have amount of the fossil fuel or the energy that is coming, 80% is coming from the fossil fuel. And day by day, as our living style is progressing, we are more dependent on the different equipments, different lifestyle. We are more used to that, which require more and more energy, and it's expected to rise up to 50% in next 20 years. And it's also given by this Intergovernmental Panel on Climate Change. They gave the report that the greenhouse gas release from this sector is about the 30% from the burning of the fossil fuel. So to control that and to overcome this crisis, there are different alternatives now people are searching. So among the alternatives, bioenergy has evolved as a quite good alternative and quite appropriate or attractive alternative to us. So because there are several advantages of use of bioenergy. So these are naturally low in sulfur, aromatics, and other toxic compounds, and that those compounds which are adversely affecting the environment or affecting finally the human health. Then bioenergy is generally produced from the biomass. So this biomass is a sustainable and a renewable source. And bioenergy are generally less toxic and it's biodegradable in nature. Also, if we are shifting from the fossil fuel energy to bioenergy, so we are moving from the non-renewable to the renewable source of energy. So that will secure our energy resource. And also it's contribute to the rural development through domestic productions. So in small scale productions also possible in some form of bioenergy. Also, it's seen that bioenergy is producing low amount of greenhouse gases than the normal fossil fuel energy. So bioenergies typically can be divided in different types. So among this are the biodiesel as their biogas. Among the biogas, we have methane, one source of energy, and biohydrogen is another source of energy. Both are coming under biogas. Then bioelectricity is there. Biobutanol is one of the form of bioenergy, bioethanol. So now researches are going on in all sector of the bioenergy. So moving to the first type of bioenergy, which is the biodiesel. So biodiesel is produced by the transesterification of the triglycerides or the common oil, which with the short chain alcohol and in presence of some catalyst. So this oil using the alcohol, it's converted to the biodiesel, and the glycerol is the byproduct from it. So this oil is coming from the different sources. So depending on the source of this, we can divide the biodiesel in several generations. Like first generation, so it started long ago, but there are certain limitations are there in that different generations. So that's why one is moving from the first to second and second to third to overcome the limitations of each of the generations. So first generation is the edible biomass. So oil coming from the edible biomass like corn and mustard oil, palm oil, etc. These are coming from that. Then second generation is the non-edible biomasses, non-edible source, that is there's different Jatropha, Pongamia. These are the certain plants which comes in this. And in the third generation, we have mainly the microbial sources. Okay, microbial sources, that's where microalgae is there, bacteria, yeast, all those are coming under the third generations. And there are still more development is there in the fourth generation, where directly the biomass is converted to the bio oil by different chemical or physicochemical means. So, and also some genetically engineered strains are used to produce the oil for production of biodiesel. So biodiesel first generation is, as I told, that it is from the fruit crops. First generation biodiesel from the oils we use, like soybean oil, palm oil, corn oil. So these are the coming from the food oils. So this when we are using this food oils for the biodiesel production, biofuel production, then it creates this problem of food versus fuel competition will come. So that's why there is the to overcome that limitation. So it's moving from the first to second generation. So second generation includes different non-edible plants like Pingamia ponata and Jatropha and other different non-edible oils are then coming into the picture. So that will solve the previous problem, but another creates another problem, that these feedstocks have very low in the oil yield. Okay, so total oil yield quite is very low, and we are not able to use the whole plant for the oil. Only the seeds are used for the oil production. So that's why, and also it's required the huge amount of arable lands. So that land competition is now came into the picture. So this lands can be used for production of fruits, for agriculture of the food. But if it is used for the biodiesel, so it's also creating a controversy in that. So to overcome this problem, now moves to the next generation, that's in use of the oleaginous microorganisms. Okay, so these microorganisms are able to accumulate the lipid of their about 20 to 50% of their total dry cell weight, depending on their nature and depending on the environment they are growing. So among these microorganisms, there are several microorganisms there. So microalgae is there, bacteria is there, yeast are there. So all of the categories of microorganisms can be able to produce the oil, so in sufficient amount. So this is the steps for the biodiesel production as shown in this figure. If it is the green plants or the algae, so they use the carbon dioxide and solar energy for the growth and production. So oils are generally extracted from these cells, and then it's used for the biodiesel production by the conversion method like transesterification. And then after this biodiesel is produced along with this byproduct, that is glycerol, is also produced. As glycerol can be utilized for production of glycerin as a byproduct with the biodiesel. So this can be used in a normal engine in our transports, and the released gas, carbon dioxide released from this, can again be utilized by this algae to grow. And so there is a complete cycle we can have. So advantages of oleaginous microorganisms are higher productivity, and also the simple upstream and downstream processing, and then flexibility in the genetic modification for the target products, and ease of cultivation in controlled environments, and also the non-dependence of the climate conditions. Okay, so we can provide a proper climate conditions, and it's not a very stringent one. So these microorganisms can be used in different climate conditions. So that will vary their oil accumulation, but not that much. See, this is the this table is showing the microbial lipid production by different types of microorganisms. So we call a microorganism as oleaginous if it can accumulate oil more than 20%. See, there are only few examples. So lot many microalgae are there, lot many yeasts are there, and bacteria is recently used in here is come in the this category as oleaginous microorganism. So this Endesmus, C. Candida, Chlorida vulgaris, and Chlorella vulgaris and Oxynochlorella protothecoides. So these are some microalgae which are used for production of the lipids. So they see the lipid content of this is 34, 89 and 66%. This is just an example, and they can be used with the substrates, which is maybe the wastewater or some different substrates can be utilized for their growth and the lipid accumulation. Next, another is the yeast cells can also be utilized as the oleaginous microorganisms. So like Rhodosporidium, torulloids, this Rhodosporidium, Traxivolivi, and Trichosporon. These are certain examples of the oleaginous yeast, and they can able to produce the lipids of 69 to 74% of their total weight, and this particular organism can produce 61% of the lipids of their total weight, and Trichosporon 53. Another is the oleaginous bacteria. This is also there. So one isolated bacteria, DS7, when they grow in the dairy wastewater, they can accumulate lipid up to 72%. Also Rhodococcus opocus, this is a very well-known oleaginous microorganism, that bacteria, which can grow in the refinery wastewater with the lipid content of 86% of their total weight, which is quite high than the previous. So that's why this bacteria is also preferable over the microalgae, algae or other organisms, because it can accumulate more amount and also utilize. So each of them has their own advantages and some disadvantages. So this table shows the biodiesel properties of this different sources. These biodiesel properties from the oil from the different sources. So this one is the from biodiesel properties of the edible oil, rapeseed and cottonseed oil. So we can see the this one. See from the Seton number, and this is the standard value. It should be the standard value given by ASTM standards for proper biodiesel, which is more more near about the diesel properties. This is the standards given. So according to that, this rapeseed and cottonseed oil, this is the edible oils, its Seton number is quite low. So its ASTM standard said the Seton number should be higher than 47. In case of non-edible oils, this Seton number is also not reaching that 47. But in case of microorganisms, we can see that Seton number is fulfilled for so. And all other points also are favorable in this case. Okay, so now another type of biodiesel is, another type of bioenergy is the bioethanol. Bioethanol is another good option for the fossil fuel. The different sources for bioethanol productions are the sugar, starch, and lignocellulosic biomass. From this, it can be produced. Like sugar cane, corn, grain, potato, wheat, sugar beets, all are exploited at the commercial level of bioethanol production. So among the, here also we have different generations of the production of bioethanol. And so first generation also here we utilize the food for the crops which used as the food that are utilized for the bioethanol production, like sugar cane and corn. So the sugars collected from these sources, and those are used for the bioethanol productions. In the next generation of bioethanol production, so lignocellulosic biomass is utilized for the bioethanol productions. The most used microorganism for bioethanol production, so this is the fermentative production of bioethanol is carried out by the Saccharomyces cerevisiae. So this is the most common microorganism we know to that produce the bioethanol. So the bioethanol productions can be divided in 3 main steps. If we are talking about the lignocellulosic biomass, so we need to do some pre-treatment of the biomass. So that's includes some physical, chemical, and biological pre-treatment methods. So this can be milling, liquefaction, saccharifications. So these are coming for under the pre-treatment of the biomass. Then next stage of this is the fermentation. So after pre-treatment biomass, so in pre-treatment, what we do, that's complex carbohydrates are converted to the simple sugars. In the fermentation, that simple sugars are converted to the ethanol by Saccharomyces cerevisiae. Now, in the next step, this purification of the ethanol is carried out by just distillation, dehydration, and co-production methods. And co-products can be separated from this. So another this bioproduction is the co-production of the biodiesel and bioethanol from the algae or microalgae. So simultaneously we can produce both. So it's a kind of biorefinery approach where two types of bioenergy can be produced from a single source. Like here algae is grown, and it's after harvesting the algae, it's after removing the water, the oil can be extracted from the algae, and that oil can be converted to the biodiesel by transesterification method, and then the remaining biomass. So we are not discarding any part of this. So remaining biomass of the algae is fermented with the yeast. We know that is the Saccharomyces cerevisiae. So we get the converted the bioethanol. So both two different types of bioenergy can be produced here from the single source. So recently different researchers are going on there. So it's here we give some example of that where we use the microorganisms. Sorry. This is the this one is the microorganisms are the Saccharomyces, and these are the algal biomass which use. So bioethanol yield are this one. So these are the different percentage of the bioethanol production from different sources. Then also in recent years we have seen that apart from the Saccharomyces, some other are also there for the productions, like this other microorganisms are also there, like Klebsiella, Thermoneobacter, Clostridium. These are the microorganisms, and these are the substrates used, renewable substrates, banana peel, or some rice straw hydrolytes. So these are the coming, and the substrates are coming under the lignocellulosic group. So we get the good amount of biodiesel yield from this. Next, the biogas. So biogas is also important, financially sustainable, ecologically safe, renewable source of energy. So it plays an important role by fulfilling like environmental change and public health depreciation. So okay, so why biogas is formed by the process of anaerobic digestion. So it's clearly audible. I had some connection loss actually in between. So now it's okay.
Yes, ma'am. It's
Okay.
clearly audible. Okay.
Thank you. So the biogas is formed by the process of anaerobic digestion methods. So there are different substrates are there for the that can be used for the biogas production, like manure, food waste, biosolids, solids, agricultural waste, and waste than the sludge from the anaerobic and from the activated sludge process. So those can be utilized for production of biogas by anaerobic digestion methods. So biogas can be produced, and this biogas can be used for electricity, heat, fuel, gas, speed. So all are these, they are different different applications of the biogas, and also the digested which obtained, which can be utilized as the fertilizer. So generally in the biogas, it's a combination of gas which contains the methane of 55 to 70% and carbon dioxides and certain minor quantities of water vapor and hydrogen sulfides, also hydrogen and siloxanes. So except for methane, other gases present in biogas are unknown, unwanted, and this biogas can be regarded as the pollutants. Okay, so these researchers going on to reduce the presence of these gases like hydrogen sulfides and other gases apart from the methane. So the major applications of methane gas as biogas are, this can be already we discussed that gas heater used for vehicles, kitchen stove. This can be used like we use biogas can be used as a cooking gas as a replacement for this what we are using right now, and after purifications used as the natural gas also. So already I discussed the different sources of biogas are this. So this are there. These are the different applications of that. Then another type of biogas is the biohydrogen. So instead of methane, the biohydrogen is produced. So hydrogen gas is defined as the energy carrier gas and is environmentally sound, colorless, tasteless, and simple component occurring in water, biomass, and fossil fuels. So at present, biohydrogen is regarded as most capable energy alternatives based on the climate change and energy utilization purpose. And so hydrogen production can be obtained by different methods. One is the chemical method, another is the biological method. So in chemical methods, it includes steam, reforming, catalytic and cracking, coal gasification, partial oxidation, etc. And in biological productions, it can be dark fermentation, photosynthetic process. These are two main. So we generally utilize the dark fermentation, which is the anaerobic part of the anaerobic digestions, and where the different bacteria or microbes are acting to produce the hydrogen from the different sources. So biogas and hydrogen are gaseous fluids. They by the biological process, it exists naturally in absence of oxygen. So this these are the this production process of biogas and biomethane and biohydrogen is completely an anaerobic process, and there are different stages of reactions which produce the hydrogen or the methane. So it's starting with the complex compounds like carbohydrate, protein, fats. Generally, what are present in the waste, those are hydrolyzed in stage 1, and it will produce some simple forms like sugars, amino acid, fatty acids from this complex sources. Now, next step is the acidogenesis, which converts this complex, simple components into the more simpler components like carbonic acids, which also known as the VFAs, volatile fatty acids, some alcohols, little amount of hydrogen and carbon dioxide. Finally, this is also another stage is the acetogenesis, where this acid is converted to the acetic acid, hydrogen, carbon dioxide, and here ammonia, H2S, etc. So other components are also produced. Finally, methanogenesis is there, where these compounds is converted to the production of methane and carbon dioxide in the reactor. So in several stages, a lot more microorganisms are acting to accomplish this all these processes, all these reactions. So initial stage, as I told, that hydrolysis, where complex organic polymers is converted to the simpler forms. So hydrolysis, this is carried out by different organisms like Clostridium, bacteroids, Buteri vibrio, Bifidobacterium, Bacillus, Streptococcus, etc. And then next stage is the acidogenesis, which produces hydrogen, carbon dioxide, and fermentation products, organic acids, short chain fatty acids or volatile fatty acids. So these are known as the acidogenic organisms, and among this there are different organisms are there, like Bacillus, Clostridium, Micrococcus, Pseudomonas, Lactobacillus. So all different organisms are present in the reactor to carry out this next stage of acidogenesis. So it's required pH of this for carrying out this particular reaction, and pH is favorable. pH is 4.5 to 5.5, and generally this stage is completed within 10 to 15 days. The next stage is the acetogenesis, where the main product of acidogenesis in this stage, and the methanogenesis is the next one. So the bacteria which play a main role in this have a long lag phase and optimum pH of 6, which converts this to the acetic acid. So other compounds to as produced to the acidosis, hydrogen, acetic carbon dioxide. So these are the microorganisms: Syntrophomonas, Syntrophobacter, then Paleomaculum, Clostridium, etc. So other bacteria play main role in the acidogenesis stage. Next stage is the final stage, we can say, is the methanogenesis. So at the end of this stage, we can obtain this methane productions, and this is carried out by this particular, these are organisms like Methanosarcina, Methanococcus, Methanotrix, etc. So in these stages, so all methanogenic bacteria come under acetoclastic categories. So hydrogenotropic methanogens are there. So that in that case, in dark fermentation, that we said, in removing the light, so the last stage can be stopped by removing the methanogenic bacteria. So in that case, the hydrogen will be the final product we get. So in the same anaerobic procedure, we can obtain hydrogen if we want, and we can add methanogenic bacteria if we want to produce the biogas or the methane. So nowadays, microalgae also can be used for this anaerobic digestion process. Okay, so biomass, microalgal biomass can be utilized here. It has several other advantages, for example, higher energy efficiency, and it does not require the oil or extraction stage, lipid extraction. Then final methane is also produced in the gaseous phase. So another type of the bioenergy is the biobutanol. So biobutanol is a more newer type of its own kind, and it is also regarded as a better one than the bioethanol. So butanol is 4-carbon straight chain alcohol, colorless, and it's a flammable. So it is a superior fuel considered as to the ethanol in terms of engine performance, and also it will not require any engine modification for efficient combustion. So in the present engine, whatever we have, so in that it can be utilized as it is without modification in the engine. Then biobutanol has some similar properties to gasoline and compares to the ethanol, like long chain carbon has, then higher volatility, polarity, combustion value, octane rating, and low corrosiveness it has, then lower heat of vaporization. So this makes a better fuel than the others, and thus making biobutanol a better option for blending with the gasoline. So already we can, and there are some researches going on where biobutanol is utilized and it's converted, it's used along with the gasoline and with the different blends. And also very importantly, this is referred as the drop-in fuel, and which can be blended with gasoline up to 85%. Even this biobutanol can be utilized as fuel for the aviation. As the aviation fuel. So biobutanol is generally produced by this particular bacteria, Clostridium acetobutylicum. So it's a completely anaerobic process, and it's the fermentation method is known as acetone butanol ethanol fermentation. So in this fermentation, all these three products are produced, and there can be modification in the environments to can be done, which can lead to the maximum production of butanol. So we need to do the distillation process and to purify the butanol from the mixture. So AB fermentations can be divided into also the two phases, then acid fermentation phase and then solvent fermentation phase. So here in this acetates can be converted with the different stages of conversions. So ethanol, acetone, and butanol is produced here from this. So in the solventogenesis part. So among another type of bioenergy is that the bioelectricity. So in the today's scenario, electricity generation and waste management from the eco-friendly process is achieving the global consideration. So bioelectricity generation from wastewater can be achieved in the microbial fuel cell. So where we can obtain the bioelectricity, so that instrument or the setup is known as the microbial fuel cells, where these microbes which present here acts as a catalyst to convert the organic waste into the electricity. So microorganisms, so there is a two chambers are there, anode and cathode. Generally microorganisms which are anaerobic in nature, so they are present here, and it will convert the organic matters to the carbon dioxide and H2. So already we seen that all the same microorganisms in there present in the anaerobic digestion system. Okay, so that that particular microorganisms or sludge can be used here to convert this organic matter present in the wastewater to hydrogen and the carbon dioxide. And so these two chambers is separated by the membrane. So semi-permeable membrane, say. So this proteins is passed through from this membrane to the cathode chamber, but it is oxidized by the oxygen to the H2O. So in this cathode chamber. So this is the reactions occurs in this chambers, and there is an external resistance is connected through which the electricity can be flown. So that using the multimeter we can measure that how much electricity is generated. So now moving to so these are all different types of bioenergy. Now I discussed. Now moving to some case study which we performed in our lab for the production of biodiesel from the isolated oleaginous bacteria from different wastes like dairy wastewater and lignocellulosic waste. So in this work, what we have done is collect the bacteria from this dairy industry that is located in the Rourkela only. So it's collected and samples, and we isolated different almost 9 different bacteria from there, and among them we did some preliminary studies for their growth and lipid accumulation in laboratory conditions. We use the media like dairy sludge extract media, and temperature is 30° pH maintained at 6.8 with and grown for 72 hours. And from the preliminary studies, we find that DS7 isolate was most highest accumulator of the lipids. So with this DS7 microorganism, or DS7 bacteria, we studied the further different experiments, like we studied that what are the different carbon sources it can utilize for the biomass production and the lipid accumulation. So we tested for this, we used cellulose, lactose, sucrose, starch, glucose, and xylose. So among this, DS7 here results are shown for 3 different isolates, so that is 1, 6, and 7. So we have seen in all the cases DS7 is to have the higher productions of the biomass as well as the lipid accumulation from this. So the lactose utilization by the DS7 was the highest. So can see that the growth is not that much high. Okay, so about biomass growth is about 5 gram per liter, and whereas the lipid content in the near the a 90% lipid accumulation is 90% of its total weight. So this biomass production and the lipid accumulation, these are the contradictory phenomenon. So when we have the highest biomass, the organism is having favorable conditions, that's why it is growing rapidly. And when there is not favorable condition is there, so there is a retarded growth or the slow growth is observed. When the microorganisms is put under the non-favorable conditions, so they try to accumulate or store their energy in the form of lipids, so we get the higher lipid accumulation. So that's these two things are contradictory, that we have the higher biomass in certain cases, so we won't have the higher lipid contents, and where we have the higher lipid contents, so biomass. So depending on that, we this results, we do some more experiments with now with the wastewater. Okay, so previously we carried out the experiment with the synthetic medium, and now we carried out with the wastewater that is collected from the same dairy industry, wastewater dairy industry. So here different wastewater combinations are used. This is first one is the raw wastewater. This second one is the dairy wastewater with presence of glucose and dairy wastewater, and just we combine the MSM previous MSM media with the dairy wastewater combined into two 2:1, 1:1, and 1:2 ratios. These are common. So from here we can see that raw dairy wastewater also showing good biomass and the lipid yields. So lipid yield almost we can see 70, more than 70%, near about 80, and the biomass also production is the 4.5 gram, 4.2 gram per liter. And so the highest, although the highest results we get in the this particular combination, dairy wastewater with MSM in 1:1 ratio. So lipid productivity also we can see that the highest in the this particular 1:1 ratio when there is dairy wastewater is mixed with the normal minimal salt media. So we also tested the time profile for the lipid accumulation and biomass production by the DS7 microorganism using dairy wastewater, in the dairy wastewater utilizing. So we can find out the biomass and the lipid content and the lipid accumulation in by this particular DS7 isolate. And here we see that the maximum lipid content is about 72% with the and the biomass production is also about that 4.2 nearby productions. Okay. So along with this, we also tried that how the dairy wastewater is changing. It's, what are the different changes occurs when it is growing. So we measure the reducing sugar in the dairy wastewater, and we calculate the biological oxygen demand in that particular wastewater. So initially it was found that glucose concentration about this much 3 gram per liter, and where the BOD is this much 2.0 gram per liter, and as the fermentation proceeds, it's almost nearby, it's going to be more or less than 0.5, and BOD is almost 200. Reducing sugar reduced to 0.5 gram per liter, and BOD is reduced to 200 milligram per liter. So after growing the microorganism, what we did is they use the microorganism for and see them in the confocal microscope that how the lipid accumulation occurs. So in this red region is showing the presence of accumulated lipids. So this one is the fluorescence phase, and this is the dark field picture. So from this picture we can see that this red zones are showing the lipid accumulation. So this bacteria is ability to accumulate a large amount of lipid from their total weight if a proper condition is provided. So in the next step, what we did, we extracted the lipid from the cells and try to characterize that lipid by different methods. Like this one is the NMR methods we used for characterization of the lipids. So there are typical peaks are present in the both proton and C13 NMR. So in that we see that presence of this peaks are showing that these are the groups present in the sample. So from this spectra, it can be said that it has been observed that DS7 bacterial lipids are primarily made of saturated fatty acids. So So after analyzing these results, we can say it's saturated fatty acids are mostly present here, and the chains of the fatty acids are nearby this C14 to C8. So we also characterize this oil but after converting it to the fatty acid methyl ester after trans-esterification process. So that means this is the proper, we converted the oil to the biodiesel by trans-esterification methods and then characterize it by this. So previous two were the characterization for the oil, and this is the characterization of the FAMES, that is the biodiesel. So this typical peaks are there for these OC bonds and CO ester. So that's we confirm that ester bond is present, which is the typical for the biodiesel, where we have seen that alcohol esters is produced for the fatty acids. Also, we characterize this biodiesel with the FAME, with the GC analysis. Okay, we also call it the fatty acid methyl esters, and we analyze with the GC, and these are the compounds found with this much percentage. And using the GC data, this is also represented in this figure in the graph. So we have you can see that the maximum presence is the C16 compounds, and also the C18 with a one double bond is the maximum. So 21% is present C16 chain ester, and this one is the C18 chain ester. So these are two the main components present in the our oil. So using that data from the this GC data, we use some empirical equations to calculate the biodiesel property of our produced biodiesel from that particular organism. So this is the degree of saturation, iodine value, saponification, cetin number. So from this we can see the cetin number is that within the range, it's matching with the limit. So it should be higher than 47. So this is having 89. So it's quite matching with the limit set for the ASTM. So it has the ability, we can say this biodiesel is a potential. This from what biodiesel we are producing from the oleaginous microorganism DS7 using the dairy wastewater has potential to be used as biodiesel. Although there are several more studies are required for your required further. So we carried out further certain some experiments where we try to find out the whether this particular organism can utilize other different wastes or not. So we in the previous case we use the dairy wastewater, and this case we use the waste, solid waste like paper cup, cardboard waste, and the vegetable waste. So these three wastes are used here for production of the biomass and the lipid by this DS7 isolate. So we find that it's producing this much 3.29 gram per liter of the biomass when paper cup, and 6.35 for cardboard, and 3.96 for vegetable waste. And see this opposite type of lipid content is produced, that is highest lipid content is produced for paper cup, then lowest lipid content where we have the highest biomass, we have the lowest lipid content, where we have the lowest biomass, we have the highest lipid content. So this also giving similar kind of results that our if condition is favorable, then biomass will be produced, then the bacteria will or the microorganism will grow faster. That means it will more emphasis, it's produced more amount of biomass. And if the condition is not favorable, then these microorganism starts storing these most of this carbon source, whatever available carbon source, as an lipid. So what we did here, we choose this from this. We choose the vegetable waste that as we find the productivity is highest. Lipid productivity is highest for the vegetable waste, and therefore we use the vegetable waste for biomass and the lipid production. By this particular, and we perform the optimization study with the incubation time, and these are the parameters we use, like incubation time, amount of waste, pH, and inoculum size. These are the parameters we used for the optimization study, and these are the labels for this optimization. Incubation time is the 72, 96, and 120 hour. Amount waste added is 3.5, 9.25, and 15, and pH is 6, 7, and 8. Inoculum size is taken as a percentage, 5, 10, and 15. So we, for the optimization study, we use the RSM, that is the response surface methodology. And in that we use the box Bacon design of experiment, and we take the help of the statistical software, that is the Minitab, to design and analyze the result for this. So using this 4 different parameters at 3 different levels, we have this, like, has given 27 total experimental runs. So we performed with the given conditions, and this is the temperature. This is the different conditions, coded and uncoded values, -1, -1, 0, and +1. So we can see that different biomass and lipid produced and lipid contents are calculated. Lipid contents are there. So different sources are we have seen, and these are the different runs are there. 20 total 27 runs. So using the model, we calculate the biomass and the lipid content. From here, the model developed one equations for the biomass production and the lipid accumulations. So now to validate the model, we did the this software is only we did the ANOVA analysis. Analysis of variance is carried out. So we see that model is having p-value 0.011 with a linear square and interaction. So all terms are less than 0.05. So p-values are less than 0.05. That means that it is having the higher, means more than a 95% confidence level is there. And also the model lack of fit is not significant, which is higher than 0.05. So that is model is properly fitting with the experimental data. So these are the model equations we obtain for biomass and the lipid content. So these two models are experimentally fitted with this experimentally fitted with the experimental results. Now we with this we optimize with the help of this particular software, and we get that optimized condition is for biomass and biomass production, and the lipid content is 120 hour. The amount of wastewater is the 3.5 gram with a pH 8 and inoculum percentage 6.31. So this is the optimized conditions we get, and after performing the experiments under this condition, we get the optimized biomass and lipid content of that this much amount. This is the 7.6 gram per liter and lipid content 87.45%, and we have the productivity of 1.449 biomass productivity with the lipid productivity of 1.267. So the percentage improve, we can see for biomass is almost 47% improve is observed for the optimized after optimization, and in the lipid content we have the 15% improvements, and in the productivity of the lipid it's almost double. So we find that this much 50% improvement is there, 50 for this percent. 50% improvement is there for the productivity. For this case also, we carried out the analysis, GC analysis, and using the GC data, we find out the biodiesel property of that biodiesel produced from the oil using that DS7 microorganism with the substrate vegetable waste. So this is the particular characteristics we calculate for this. So here also certain number you can see that quite high. So we have in previously slides where we discussed the general biodiesel production from the different oils, plant oils. So then that as certain number is quite low. For in that case here we have seen the certain number is quite high and more near to the standards given for a biodiesel. So in conclusion, we can say that this particular strain DS7 is an oleaginous microorganism bacteria which is able to produce the lipids more than and accumulate the lipid more than 18% when different grown on different carbon sources like lactose, sucrose, starch, and it can efficiently utilize the nutrients present in the dairy wastewater, and it can able to accumulate lipids in that case up to 72%, and the properties of accumulated lipids is suggesting that it has a potential for the being used as biodiesel. Now also the isolate able to grow and utilize the vegetable waste. So not only the dairy waste, but also it's tested for utilizing the vegetable waste for biomass and the lipid productions. There also it shows 87% lipid accumulations and 7.6 per liter biomass productions with a high productivity of the lipid production. So I discussed the work, which is this two publications we had recently. From these two publications, this awards is described here, and these are the certain references I used. So this is the my team who are working currently in my lab, and some of them have already passed out. They're doing MTech and BTech, so they carried out these works I presented here. So thank you all audience and the coordinators who gave me the opportunity to speak here, and we hope we can move to a sustainable society by having more of the bioenergy in our day-to-day life. We can utilize them, and we can move to a healthy, sustainable.
Ma'am, you are not audible right now.
Hello, Dr. Dotto, you are not audible now.
Hello.
Yes, Dr. Kosturi Dotto, I think, ma'am, you are not audible. So, Sinjini, please proceed.
Okay, so thank you so much, ma'am, for this insightful presentation. And I would now like to do a little announcement, which is for all the participants here. The feedback link will be provided in the chat box shortly towards the end of the session. And please fill it up, and the e-certificates will be generated and sent to respective email IDs within a week. I repeat, within a week. Coming to the end of the session, I would now like to call our principal, sir, Dr. Amit Shankar Datta, take over and conclude the session. Sir, are you here?
Hello.
Yes, sir.
Am I
Please carry
audible?
on. Yes, sir, you are audible.
Thank you, Kosturi Dotto, for your valuable research experience. Yeah, you have worked in a very latest field of biotechnology, that is biochemical engineering and bioprocess. Because nowadays, most of the latest in research going on all over the world, especially in the Germany or this on this field. So the fuel problem or the gas problem, these are all under this category. So by this exchange of views and your work, our student and faculties will be enlightened. So in near future, when we are arranging such things, we definitely make a contact with you. And thanks for your valuable time you are sharing with our institute. And thanks you and the conclusion.
Thank you so much, sir. Now coming to the last part, I, on behalf of Eminent College, would like to thank our speakers, both our speakers, the chairman of the webinar, Dr. Amit Shankar Datta, the convener and moderator, Dr. Kaushik Biswas, technical coordinators, Mr. Shirojit Gun and Mr. Moyuk Sarkar, our coordinators, Ms. Ankita Bonik and Mrs. Amrita Das, and all the participants who are present today to make this webinar a success. Saying this, I would conclude and end the webinar. Thank you so much. Okay, the feedback link has been provided in the chat box. Please fill it up, and the e-certificates will be given within a week.
The feedback link has also been sent to our official Telegram group. So access our Telegram group for the feedback link and fill it up for the certification. Okay.